I'm Daniel Levine, and this is the Bio Report. Small molecule drugs and monoclonal antibodies often fall short at addressing severe inflammatory and immune-mediated diseases. Mesoblast has spent more than 15 years industrializing mesenchymal stromal cell therapies to treat these conditions. In late 2024, it won U.S. Food and Drug Administration approval for Ryoncil, the first mesenchymal stromal cell therapy approved in the United States. Ryoncil is approved to treat steroid refractory acute versus host disease in pediatric patients 2 months of age and older. Mesoblast founder and CEO Silviu Itescu discusses how this class of therapy can help dampen cytokine-driven hyperinflammation, how the company is extending its platform into other high-burden conditions, and what distinguishes its second-generation cell therapy now in development from its first. Silviu, thanks for joining us.
Nice to be here.
We're going to talk about Mesoblast, its development of off-the-shelf stem cell therapies for autoimmune diseases and other conditions, and its approved product to treat graft versus host disease. Let's start with some basics. Mesenchymal stromal cells are at the core of your allogenic therapies. For listeners not familiar with these cells, what are they and how do you obtain them?
Sure. We've been developing these technologies for 15 years now and have established very precise manufacturing processes we obtained from healthy donors, bone marrow material that allows us to extract very rare cells called mesenchymal precursors. And once we extract them, we have processing capabilities that allow us to expand them in very large numbers. The unique aspects of these cells is that they can be expanded to large numbers. They don't activate the immune system and therefore can be used from one healthy donor to treat thousands of unrelated recipients. So through this manufacturing process, which is now industrialized, we generate very pure populations of these cells that are well characterized, that have got potency measurements that ensure that every vial of cells is identical from one vial to the next, and that we end up putting these cells into little vials that are then frozen at minus 130 degrees and then shipped from our warehouse to the hospitals and the end users through what's called cold storage facilities and supply chain. So the whole process is highly industrialized, well characterized. It's well regulated, safe, demonstrated potency, and we can show that each vial is the same as the next, and that's critical to ensure that the product is dependable, safe, and meets FDA requirements.
You founded Mesoblast in 2004 after an academic career. What convinced you to leave academia and build a company around these cells?
Well, I was head of transplant immunology at Columbia Presbyterian. I was involved in overseeing inflammatory treatments, immune treatments to moderate the immune system in patients with heart transplants at Columbia, largest transplant center across the U.S. And then we were looking for alternative therapies that could be used to help patients with advanced heart failure because of the very limited supply of organ donors for heart transplants. And we had quite a breakthrough result that showed that these type of cells could be used to repair and rebuild blood vessels and heart muscle. In animal models, we published that, and that led to founding Mesoblast on the basis that I wanted to take this type of technology and scale it up and evaluate it in patients with severe advanced heart failure. And here we are, these many years later, we've commercialized a platform technology that has the advantage of targeting inflammation in very severe diseases, including inflammatory heart disease, and is able to be scaled, and through phase 3 trials has demonstrated evidence of efficacy and safety across multiple very severe diseases that are otherwise not addressed by either small molecules or monoclonal antibodies.
What problems are you trying to solve in inflammatory and immune-mediated diseases with these cells now?
Well, there are quite a few diseases. Inflammation is at the core of, and inflammation is caused by multiple arms of the immune system. And so it's not a surprise that small molecules or antibodies that target just one pathway are not very effective in turning off severe inflammation. And we saw that with COVID and COVID respiratory diseases, which was the prototype for a cytokine storm. And so those diseases that have these very bad inflammatory components progress, are not responsive to these drugs, and have high mortality. So graft versus host disease is one of those very bad diseases. It's a complication of a bone marrow transplant where the recipient sees the donor graft as foreign, and the graft actually sees the host as foreign as well, and the graft attacks the host with multiple immune systems and multiple arms of the immune system and multiple cytokines. And to date, other than our MSCs in children, our mesenchymal stromal cell Ryoncil in children, no other drugs have demonstrated effectiveness in children under the age of 12. And that's testament to the difficulty in managing diseases that have a lot of inflammation with traditional medicines. And so the mesenchymal stromal cell is uniquely able to be activated by these multiple arms of the immune system and turn them off. So graft versus host disease in children, we have the only product that's approved to treat these children under 12, a disease that otherwise causes mortality rates north of 70, 80% without our cells. And with our cells, we have 70% responder rates and survival that is both short-term, and now we have data that says we have a lot of children who are surviving through 5 years, a point at which clearly the disease is well controlled. And so beyond graft versus host disease, a number of other disease states have high mortality and high morbidity due to the severe inflammation that accompanies them. And we're targeting inflammatory pain and inflammatory cardiovascular disease, in addition to the graft versus host disease in children and then in adults.
Well, what is Ryoncil? Is it just a purified delivery of these cells, or is it contain anything else in it?
Ryoncil is a very pure population of these mesenchymal stromal cells, highly enriched and expanded and concentrated, so that each vial of Ryoncil is well characterized, contains 25 million cells of these mesenchymal stromal cells. And so we dose in patients by weight. Our dosing is 25 million cells, 2 million cells per kilogram. And so however many vials of 25 million cells an individual needs, they're being offered as an IV infusion. And some small children get only a few vials. Larger children will get a lot more vials at 2 million cells per kilogram. But those cells are very well characterized, and each vial contains 25 million of these potent cells.
At first glance, it may seem counterintuitive that you would treat a patient who is having an adverse reaction to a stem cell transplant by giving them stem cell derived therapy.
Well,
what does these cells actually do?
Well, that's a problem with the terminology stem cell, right? It's a catch-all phrase. The the stem cell transplant that children and adults get for bone marrow cancers is bone marrow transplant that contains what are called hematopoietic stem cells. And those are the stem cells that rebuild your bone marrow elements. And they're important because the chemotherapy that's used to treat the underlying leukemia has wiped out the bone marrow. So you need to repair and rebuild the bone marrow with those type of stem cells that cause that are the precursors of white blood cells,
Right,
red
but
blood
what
cells.
are these cells
So
doing
these
to
cells,
calm the immune
these
system?
these cells have nothing to do with hematopoietic cells. These cells are immunomodulatory lineage cell therapy. And what these cells do is they're activated specifically via receptors that they have on their surface by the inflammatory mediators that are produced by the immune cells within the graft. And those cytokines activate our cells so that they then release anti-inflammatory factors that turn off the inciting inflammation. And so they're signaling cells that we all have in very small numbers in our marrows and in various tissues. Their job throughout our life is to control aberrant inflammation. That's what they do in our bodies. And so we expand those cells, we give them back from one donor to unrelated recipients, and they modulate immune responses. In this case, in GVHD, the immune response they're modulating is of the foreign graft. In other diseases, they modulate inflammation within the heart, within the intervertebral disc or other joints, within the gut in patients with inflammatory bowel disease. So the way they're used is dependent on the particular disease state, but the mechanism by which they turn off inflammation is common across all these different disease states.
And Ryoncil is described as donor agnostic and doesn't appear to provoke a clinically meaningful immune response. Is that just something to do with the nature of these cells, or do you have to do anything in the manufacturing process to prevent the development of an immune response?
No, these are unique cells in the body that, by the nature of their characteristics, they don't activate the immune system. They don't have certain molecules like HLA class 2 molecules on them that are normally other tissues have that shows them as being foreign. So that, and in addition to that, they secrete immune factors that protect themselves against attack from other cells. So they're uniquely positioned to not activate the immune system, therefore can be used from one donor to treat many unrelated recipients.
And what's known about them from the studies you've done to date on Ryoncil?
Well, Ryoncil in graft versus host disease in children was highly effective, and we were successful in a phase 3 trial to demonstrate that a disease that in what's called steroid refractory disease, advanced disease in children, that otherwise has a mortality rate approaching 70-80%. In that disease, we had a 70% responder rate at 1 month after multiple infusions. And those children who were alive at 1 month were free of graft versus host disease for 5 years, actually. Only 14% of children treated with Ryoncil succumbed to GVHD over a 5-year period of follow-up. So early response predicts long-term survival, which is terrific. It's a change, it's a paradigm shift in the treatment of this terrible disease in these children. But in addition to that, we have data to show that in adults who have severe refractory GVHD, not responding to the only approved product in the field in adults, and who otherwise would have a 25% survival, those type of adults under expanded access, we've shown over 60% survival with our cells, and are now we are beginning a phase 3 trial in adults where the cells are being used in second line after steroids as part of the regimen in severe disease to see if we can turn off the same disease we're turning off in children in adults with severe refractory GVHD. So we're pleased with what we've seen so far in children. We're pleased with what we've seen in expanded access in adults. We hope to be able to extend the label from children to adults. And in the meanwhile, the product in children is being commercialized, it's being sold, and it's being adopted by many centers across the U.S. Beyond,
Well,
beyond, yeah, sorry.
no, expand on that. Well, what has the commercial adoption been like?
We're very pleased. We launched this product 9 months ago, and we're very pleased by what we've seen. We've laid out the 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 value proposition to hospitals and to families. We've laid that out to insurers and to Medicare, Medicaid, and 95% of commercial carriers now have insured the product fully. Medicaid uh, covers the product fully under a particular J code and has mandated that every state in the US cover the product. Um, we penetrated about 20% of the addressable market, and we expect that we can move this and grow it to about 20%, to about 40% of the addressable market, and that's what we're aiming to do. So that's by any measures, a very successful launch and commercialization over the first 9 months. And we're pleased, and we're seeing the same sort of results in the real world as we saw in the phase 3 program.
You mentioned you've been able to industrialize manufacturing. From a cost of goods standpoint, how do you see therapies like Ryoncil competing against biologics and other advanced therapies in terms of gross margins once you're at commercial scale?
Yeah, we're already at gross margins that are at least as good, if not better, than other biologics like monoclonal antibodies, and that's a unique feature to our type of cell because it's highly scalable and it's allogeneic, and meaning it can be made off the shelf from one donor to treat thousands of people. So we're already at 80% to 90% margins. We additionally have proprietary technologies that are now being implemented in our manufacturing sites to be able to increase yield by 5 to 10-fold per unit time, and that increase in yield is what we need to address the larger volume indications such as inflammatory back pain and inflammatory heart disease. Those are not orphan indications; those are large volume indications. So we are improving our yield through proprietary growth factor-based media, as well as moving into bioreactors, which will allow us to, again, another step growth in yield and reduction in FTEs per unit time.
And how broadly applicable do you think the therapy might be? Is this a pipeline in a product?
It's definitely a pipeline and a product. The question becomes then strategically, given that these are not cheap medications; they're highly sophisticated and expensive medications, and so this technology will be generally targeting severe diseases that have got either major impact on hospitalizations and morbidity or major impact on mortality, diseases that are, even if they are higher volume, they're refractory to traditional medicine. So in cardiovascular disease, for example, we're targeting severe heart failure patients who, in an early, moderate to severe heart failure, in an earlier study we showed in a randomized controlled study that we could reduce heart attacks and strokes by about 80% on top with a single injectable on top of maximum standard of care over a 3-year period of follow-up, and together with a reduction in mortality of 60% on top of maximum standard of care. Pretty remarkable data, and those have been published in very prominent peer-reviewed journals. We will be pursuing approval for that indication in the most extreme patients with end-stage disease being kept alive on artificial hearts, and performing further confirmatory studies in the more moderate populations. But that's an example of the larger volume populations where this technology has a major role to play. But nonetheless, it's not for everyday use. It'll be for those patients with the highest risk of mortality. Similarly, in inflammatory back pain due to degenerative disc disease, there are large numbers of patients who are suffering terribly because traditional medicines are just not making a difference in terms of their severe pain and disability. And of course, the opioid epidemic across the U.S. is a result. At least 50% of prescription opioids are because of severe refractory back pain. So this is a major unmet need, a major population that needs something other than opioids, of course, and other than surgery and interventions. And in that patient population, in the first phase 3 trial of 400 patients, we showed that we could significantly modulate pain relative to controls over a 12-month period, and those who responded at 12 months remained in a reduced pain state, often in remission from pain, for as long as 3 years. A confirmatory phase 3 trial is now well underway, and we expect to complete enrollment in this second trial this quarter, with a 12-month primary readout as the endpoint that the FDA has said will support approval if we achieve the same reduction in pain in this trial number 2 as we did in trial number 1. So again, very large volume indication, but the patient population is not mild pain, it's severe refractory unremitting pain for whom there are no other alternatives. So you can see that we're developing these therapies for very bad, severe diseases, even though they may be large in number, but the unmet need is really significant.
Well, what's the thinking as you look at other indications to expand into, beyond the size of the market? How do you think about where there might be a most appropriate therapy?
Well, the way we think about this is label extensions and new indications for Ryoncil versus new indications for our second generation product, rexlemestrocel. So Ryoncil is our flagship product. It's approved by the FDA. It's used for intravenous injection at the moment in the most severe diseases like graft-versus-host disease. I expect that we will increase its use in other rare diseases, both in children and in adults, where pricing will remain at a premium because of the severe unmet need in these rare diseases where inflammation is so dominant. With our second generation product, rexlemestrocel, it's an injectable into the tissues. So it's injected into the left side of the heart. It's injected into the intervertebral disc directly. It's injected into the knee joint in people who've had ACL tears. And so it's a different concept of a product rather than intravenous, and it's being used in more higher volume disease states and therefore needs to be manufactured in a way that gives us greater yield and greater volume and lower cost of goods. And so I see that second product as the one that will open up a number of larger volume indications, whilst the first flagship product will continue to be developed for ultra rare or rare diseases in both children and adults.
And is the second generation product something other than purified cells?
The second generation product is isolated using monoclonal antibodies. So it's isolated in a very different way to give it even more potency, and it makes the product even more selective, and allows it to be modified in certain ways, both through the manufacturing process, but also genetically, potentially, to achieve even greater potency at the local tissue level.
You went through a hard time with the FDA to get Ryoncil approved. It was rejected in 2020 and again in 2023. How challenging was it to have the wherewithal to make it to an approval, and what did it take?
Well, when you have the strength of character to believe in the technology and in the data and in the results, you push forward. And our results were clear cut. We had very strong clinical data, very strong manufacturing and regulatory data. First time that we went to the FDA, we had a 9-1 vote in our favor by the advisory committee panel that the FDA had actually selected to review our data. So the experts recommended that we should be approved, and nonetheless, the FDA decided to vote against it, decided to go against it. So when you know the strength of your own data and you know the strength of your technology, you have to be resilient. And I think we've demonstrated our resilience, and we kept going back and we kept showing them more data. We showed the FDA that these were not short-term clinical outcomes, but rather 5-year clinical outcomes that were impressive. We showed them that we knew how to manufacture, and we could demonstrate that we had assays that showed that the product, every vial of our product is the same and consistent, reproducible, and potent. And so we convinced them by the nature of our science and by our resilience. And I think that's what you got to do. If you have a product that works, you have to stick with it.
And was anything learned from that process that you're now applying as you get ready for the second generation of product?
Sure, we've learned exactly what the FDA wants to see in order to be satisfied, both clinically and at a manufacturing level, and we're applying all of the knowledge that we learned to the manufacturing, to the scalability, to the testing of our product. The rigor that is going into all of our products now is the end result of the lessons that we learned through the struggles of the flagship product, Ryoncil.
And are there lessons learned for broader biotech and cell therapy companies from Mesoblast's experience?
Look, I think every technology is different. I think every company is different. I think, it's very hard to talk about applying lessons across broadly across an industry. But the lessons that we learned, we're at the forefront, the first ever adult allogeneic stromal cell product approved. It's always difficult when you're the first in class. And so you pave the way, and then the learnings from that really very much depend on the specifics of the technology of the situation. But I think a company that doesn't learn from its own missteps will never succeed, right? So I think the applicability of what we've learned about our technology and how to manage it and how to interact with the FDA is probably applicable more broadly in biotech. But I think the specifics are specific to what we've learned and what we do.
Silviu Itescu, founder and CEO of Mesoblast. Silviu, thanks so much for your time today.
Thank you. I appreciate being with you.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.